Ready for another deep dive? Let's jump into the microscopic world inside our cells. Today, we're focusing on a protein, ZBP1. It stands for Z-DNA binding protein 1.
Yeah.
Now, I know that might sound a little intimidating, but this tiny protein plays a huge role in keeping our cells running.
Right.
We're talking immunity, stress responses, maybe even fending off viruses. And luckily for us, we have this 2006 research article to help unlock some of ZBP1's secrets.
Absolutely.
So to understand ZBP1, we first have to meet its partner in crime, Z-DNA. Now, we usually picture DNA as, that iconic double helix.
Right, yeah.
Like a spiral staircase.
Yeah.
But there's a twist.
Yeah, so we usually think of DNA as a right-handed helix.
Okay.
Kind of like, a standard staircase, always twisting in the same direction.
Yeah.
And scientists call this bDNA.
bDNA. Got it.
But what's fascinating is that DNA can actually twist in the opposite direction, a left-handed helix.
Oh, wow. Okay.
So it'd be like that staircase suddenly changing direction, kind of flipping the script.
Okay.
And that's Z-DNA.
So it sounds like Z-DNA is more than just DNA doing a handstand.
Yeah, exactly. It's definitely more than just a quirky DNA trick.
Yeah.
Scientists believe that it plays a key role in how our genes are actually regulated, how our bodies respond to threats, and maybe even how we could combat viruses.
Wow. So it's like discovering a hidden room in that DNA staircase. That's a great analogy.
Yeah, exactly.
So where does our friend ZBP1 fit into all of this?
Well, this research really digs into how ZBP1 actually binds to this elusive zDNA.
Okay.
And get this, the researchers found that ZBP1 has not one, but two zDNA binding domains. Yeah, so you can picture it as ZBP1 having two hands, and each one of these hands can grab onto that left-handed zDNA.
Okay.
And they call these binding domains ZA and Z.
ZA and Z, so two hands. So ZBP1 can bind to zDNA with either one hand or two. What's the difference?
That's a great question. That's what they wanted to figure out. And the research actually found that ZBP1's grip on the zDNA is much stronger when it uses both hands together.
Interesting.
Yeah, it's kind of like realizing that our acrobat isn't just clinging on for dear life.
Yeah.
But they might be a highly trained performer actually using both hands to kind of expertly maneuver on that twisting zDNA staircase.
Okay.
And this two-handed binding could mean that ZBP1 is involved in way more complex cellular processes than we previously thought.
Wow. So we're not just talking about a simple on-off switch here. This two-handed grip could give ZBP1 incredible control, maybe even influencing which genes are turned on or off or how cells react to, those environmental threats we were talking about.
Exactly. It's like discovering that ZBP1 isn't just a key, it's a master key. It's capable of unlocking this whole range of cellular responses. But it gets even more interesting.
Oh.
Yeah. The researchers also discovered that not all ZBP1 proteins are created equal.
Okay.
They found two main versions or isoforms of ZBP1, each with, slightly different structures.
Oh, ZBP1 twins. Tell me more about these two versions.
Okay, so think back to those two hands we talked about, the ZA and ZB domains.
Okay.
Well, imagine one twin is missing a hand, and that's our second ZBP1 isoform. It lacks that first Z domain.
Okay, so one ZBP1 has both hands, and the other one is, let's say, single-handed.
Right.
Does this difference actually change how they function within the cell?
That's exactly what the researchers wanted to figure out. And think of it this way. Just like a city has different neighborhoods, right? Like you have places for work, for leisure, for shopping.
Yeah.
Well, cells have distinct neighborhoods too.
Okay.
And these are specialized areas where different cellular processes happen.
So like designated zones within the cell.
Precisely. And it turns out that our two ZBP1 isoforms, even though they're in the same city, they hang out in different neighborhoods.
Interesting.
Yeah, the full-length, two-handed ZBP1 seems to be more of a social butterfly, spread throughout the cell.
Okay.
While the single-handed version without that first ZA domain, they tend to cluster together in larger groups.
So almost like they have different social lives because of that missing hand.
Right.
But what's the significance of these different locations? Do we know why they choose these different cellular hangouts?
Well, the research suggests that those locations actually point to unique roles for each ZBP1 isoform.
Oh, okay.
Think about it. Different neighborhoods often have, they specialize in different activities.
Right.
And in this case, those distinct locations seem to be linked to how cells respond to stress.
Okay, you have my attention. We're talking about cellular stress, right? Like when I'm up against a tight deadline and my cells are feeling the pressure too.
Exactly. Just like us, cells has ways of dealing with stress.
Okay.
And when a cell is stressed, it forms something called stress granules.
Stress granules.
Yeah, you can think of them as like designated safe zones within the cell for when things get a little rough.
So stress granules, like emergency shelters for the cellular world. I like it.
Yeah.
So where do our two ZBP1 isoforms fit into this whole stress response?
This is where it gets really interesting.
Oh.
The research found that when a cell is stressed, that full-length ZBP1, the one with both ZA domains, it rushes into these stress granules.
Wow.
Yeah, it's like it's hardwired to be a first responder to emergencies.
Wow, talk about being on the front lines. Full-length ZBP1 sounds like a true cellular hero, ready to tackle whatever's causing the problem.
What about the single-handed version? Does it join the party in the stress granules too?
Surprisingly, no.
Oh, okay.
The single-handed ZBP1, the one without that first ZA domain, it doesn't seem too bothered by the stress.
It mostly stays put in its own little groups, away from all the action in the stress granules.
Hold on, so the full-length ZBP1 is rushing to the scene, and the single-handed version is chilling out in a different neighborhood.
That's fascinating. It seems like such a small difference, that missing ZA hand, but it really affects how these two versions behave during, like, a cellular crisis.
Yeah.
Knowing your interest in the listener's area of interest, this kind of functional difference within a protein family might be particularly intriguing.
Yeah, you're right to key in on that difference. And while the research didn't pinpoint the exact function of the single-handed isoform, it did offer some really intriguing hints.
Okay.
Remember how we talked about those cellular neighborhoods?
Yes, those distinct areas within the cell where specific processes occur. Well, the single-handed ZBP1, with its tendency to cluster together, it might be involved in regulating other vital cellular processes.
Okay.
Things that need to happen, even when the cell isn't under immediate threat.
Okay, so while the full-length ZBP1 is like a first responder, the single-handed version might be more like, say, a city planner or an engineer, making sure everything runs smoothly in those other vital neighborhoods, even amidst the chaos.
That's a great analogy.
Thanks.
And just like in a well-functioning city, you need both, right?
Right.
You need the emergency responders and those who maintain the infrastructure for everything to work in sync.
Yeah.
And in this case, the research actually found a fascinating connection between the location of those single-handed ZBP1 clusters and the cell's internal transportation system for genetic information.
We're talking about how DNA's instructions get put into action.
You got it. It's a pretty complex process, but to simplify it, DNA's instructions, they need to be copied into a messenger molecule called RNA.
Okay.
And this RNA then carries those instructions out of the cell's nucleus, where the DNA kind of resides, and into the cytoplasm, where proteins are made.
So it's like sending a text message with instructions instead of delivering a handwritten letter in person. Much more efficient.
I like that analogy.
Thanks.
And just like with text messages, you need a reliable network for delivery, right?
All right.
And that's where the single-handed ZBP1 might come in.
Okay.
The research suggests that those clusters that it forms, they could act as sorting centers or checkpoints along this cellular transportation route.
You're saying it's like a postal service or a distribution hub for those all-important RNA messages.
Precisely.
Wow.
And by controlling the flow of these messages, the single-handed ZBP1 could influence which proteins are produced and when, ultimately impacting a wide range of cellular functions, even when there isn't some big stress event going on.
This is blowing my mind. So not only are we talking about ZBP1 potentially, defending our cells from viruses, but one of its forms might also be responsible for managing the flow of genetic information within the cell, acting as like a conductor for the orchestra of cellular processes.
That's what the research is suggesting.
Wow.
And it's a testament to, the incredible multitasking abilities of even the tiniest proteins within our bodies.
Wow. Talk about a dynamic duo. But it doesn't end there. The researchers also explored the possibility that ZBD1, in both its full-length and single-handed forms, might play a role in another critical cellular process. Programmed cell death. Programmed cell death. That sounds a bit ominous. Are we talking about cells making the decision to self-destruct?
Well, it's not as dramatic as it sounds. Programmed cell death, or apoptosis, is a completely normal process.
Okay.
It's essential for our bodies.
Okay.
It's more like a controlled demolition than a self-destruct sequence.
Okay, so less like a dramatic exit and more like a carefully orchestrated retirement plan for cells.
Exactly. Cells have a life cycle, right? Just like any living organism.
Right.
And just like us, sometimes they get to a point where They're just not functioning at their best.
All right.
Maybe they're old, maybe they're damaged, or they could even be, potentially harmful to the bigger organism.
Okay.
So to keep everything running smoothly, the body has this, incredible mechanism for getting rid of those cells in a safe and controlled manner.
Yeah.
And that's apoptosis.
So it's like the cell is tidying up and making sure there's no clutter, no dysfunction. That's pretty impressive.
Yeah. And what's even more fascinating is that the researchers in this ZBP1 study, they found some really cool evidence suggesting that our two-handed protein might be a key player in this cellular cleanup crew.
Really? How so?
Remember how we talked about Z-DNA forming under certain conditions?
Yes.
Well, one of those conditions might actually be when a cell is stressed or damaged, beyond repair.
Oh.
It's like a signal flare goes up saying, hey, it's time for me to, bow out gracefully.
So Z-DNA could be like a cellular distress beacon.
Right. And because ZBP1 is so good at binding to Z-DNA, it's in a prime position to recognize these signals and then potentially trigger that apoptosis process.
Interesting. So it's almost like ZBP1 is the cell's quality control manager,
Yeah.
making sure everything is running smoothly, and then initiating those controlled demolitions when necessary.
That's a great way to put it.
Thanks. This whole picture is incredibly intricate. We've learned that ZBP1 can bind to this rare Z-DNA with one or two hands. It exists in different forms with different roles within the cell, and it might even play a part in this life or death decision of programmed cell death. It really highlights how amazingly complex our cells are, what's happening every second of every day.
It really does. And for me, the most exciting part is how much more there is to discover. This research, it gives us this little glimpse into the inner workings of ZBP1, but it just opens up this whole new avenue of questions about what it might be doing in other cellular processes.
It's like we've only just scratched the surface of understanding this tiny protein.
Exactly.
And its impact on, our health and well-being.
And that's what makes this field so compelling. We're constantly pushing the boundaries of knowledge, and each new discovery, it brings us a little bit closer to unlocking the secrets of life itself,
Well, I think we've given our listeners a lot to think about today with this deep dive into the world of ZBP1. It's amazing to consider that something so small, this tiny protein, can have such a huge impact, on how our cells function, how they respond to stress, and even how they manage their own lifespans.
Pretty remarkable.
What stands out to you the most from this research?
What fascinates me is how this research really challenges some of our previous assumptions about how cells actually work. We tend to think of DNA as this static blueprint.
Right, yeah.
But the discovery of Z-DNA and proteins like ZBP1 that interact with it in such specific ways, it just highlights how dynamic and responsive our genetic code actually is.
It really does. It changes how we think about how our genes are regulated and how our cells adapt to the environment around them.
Exactly. It's a good reminder that the microscopic world inside us is just this hive of activity, constantly adapting and responding to keep us going.
And as scientists continue to explore this microscopic universe, who knows what other incredible discoveries await. It's a really exciting time to be, diving deep into the world of cellular biology.
Absolutely. There's so much more to learn, and I, for one, can't wait to see what the future holds.
Well, on that note, we'll wrap up this deep dive into the world of ZBP1, but we'll leave you with, hopefully, a newfound appreciation for just how elegant and complex your own cells are. Until next time, keep exploring.